So our next speaker is Angel White, who is Dr. Angel White, who is a professor at the University of Hawaii. And I didn't memorize her bio, so I will read it from the monitor here. Angelique E. White is an associate professor of oceanography at the School of Ocean, Earth, Science, and Technology. She joined UH Manoa's Center for Microbial Oceanography, Research and Education in 2018. White's research focuses on plankton biodiversity, productivity, and elemental cycling in upper ocean ecosystems. She is currently the lead principal investigator for the Hawaii Ocean Time Series, HOT, program, and is passionate about spreading the gospel of HOT and the need for sustained ocean observations. Her 2020 TED Talk at the National Academy of Sciences was deemed one of the 20 most popular TED Talks in 2020. White has participated in more than 40 oceanographic research expeditions to sites spanning the subtropics to the Antarctic, received research grants totaling over $23 million, and published nearly 100 articles in peer-reviewed scientific literature. She was a recipient of the Alfred P. Sloan Fellowship, the American Geophysical Union Ocean Sciences Early Career Award, and the Association of the Sciences of Limnology and Oceanography Jens Schindler Early Career Award. Thank you, Angel, for agreeing to speak with us, and I look forward to this. Turn on the camera. Share this screen on Zoom.
Okay, thank you guys so much for your time, for taking time out of your morning to listen to a little bit of science. I'm not going to take too much of your time. I want to share a little bit, as you said, about the Gospel of HOT, the Hawaii Ocean Time Series. It is about time. Station Aloha, Anita introduced Aloha quite well, but it also means a long-term oligotrophic habitat assessment. This is a gathering place for discovery, for education, for scientific collaboration, and I hope what you'll get out of this talk, ultimately, it's about technology and about all of you here at the table. So here's a little snapshot of some of the components of HOT. I'm going to walk through this. This was begun in 1988, October of 1988. We have approximately monthly cruises to Station Aloha. Doesn't always work that way. I know UNOLS is still digging out of the pandemic. We have more than 50 core measurements. It is. That's okay. We have a special focus on carbon, nitrogen, and phosphorus. These are the elemental building blocks that form all of life. And we also want to try to understand microbial community dynamics. This is the invisible world. Importantly, this has just been renewed for another 5-year cycle. This is huge. This is a $10 million grant, plus 50 days of ship time. So we take this responsibility quite seriously. This is a huge investment in taxpayers, and we are trying to discover how the ocean works, how it's structured, and importantly, how it's changing. Metrics that the National Science Foundation cares about, our funder, are in the top right. Over 800 publications and a massive amount of training, 60 master's thesis and PhD dissertations. Let's see if this will work. This is the main component, so the K. Thank you, Jim, for the 2 replacements. We deploy sediment traps. We make productivity measurements. We use radioisotopes. We look at the microbial community structure. That's a colonial ciliate, one of my absolute favorites. And we do this in the mission of trying to understand the North Pacific subtropical gyre, the largest ecosystem on our planet. That is no small feat. Some components of this program beyond the core, and the core is the shipboard science. This is the Watts mooring. There's Tully Rohr looking quite smug at the successful recovery. Tully was a tech for OOI for a while, so he's got an enormous amount of experience that he's brought to this program. This is a pretty cool mooring, not just because we'd make a few passes to fish every time we go and collect samples, and that's been pretty successful, but we also look at subsurface heat, freshwater fluxes, temperature, salinity, velocity, hourly data back to 2004, from the sea surface to the sea floor. So shipboard science, moorings, and we also partner with the Aloha Cabled Observatory. This is ACO. This is the deepest operating cabled ocean observatory in the world, 4,728 meters. I think that's kind of amazing, right? I mean, you guys all understand the challenges of getting to the seafloor, let alone having instruments operating continuously on the seafloor, which is what you see in the right. We have acoustics, video, temperature, salinity, pressure, current measurements since 2011, as well as dissolved oxygen and fluorescence and turbidity. But the real most important component of this program is people. These are some of the folks that originated the program. It's Karin Bjorkman on the top left, John Doerr. They're both still with the program, so they've been with it since the beginning. On the bottom, we've got Chris Sabine. He's the ADR for our UH, so he's moved up into admin. And you can see a very unsafe recovery on that bottom right. There's some tripping hazards, right? So we've evolved since that point. But really, this is the important part here. So we keep track. We like anniversaries. We like celebrating things. And so what you'll see on that top right are our top 10 cruise participants. And on the left, we've got the top 5. So Dan Sadler is the chief scientist, 252 cruises. Fernando Santiago Mandujano, 230 cruises. Lance Fujiaki, 205 cruises. Karin Bjorkman, 167. And Blake Watkins, 187, but 110 in a row, right? It's amazing. So these folks are really the heart of this program, and they have led opportunities for education and training and research, and they facilitate the science that I'm going to share with you. As I said, we like celebrations. This was Hot 300. Some of the folks out on this cruise, some of you guys might remember Lawson, also Chris Wynn. These folks were on Hot 1, and they're on Hot 300. We're coming up to another celebration, Hot 350s around the corner. So opportunities, education, research. We've had over 400 PIs participate on these cruises, over 4,000 researchers and postdocs from around the world, not just Hawaii. We have graduate students, about 1,000 of them. Again, we've got Carolina on the top doing array recoveries. We've got graduate students doing volunteer work. There we got Blake and Juliana, one of our favorite techs that used to be on the KM. She's now in the FAL Corps. And we recognize that it is an ecosystem, right? It takes an entire village, from the galley to the crew, to the bosuns, to the captain, as well as the technicians and the science, to get the work done that we're about to show you. But these people are icons, all of you. You guys are incredibly important to the science that gets done. And I would like to say that we'd like to have like trading cards, right? Collectibles for the best of the best. So I'll start with Carolina, right? She's over 50 cruises. She's a mom. She's an analytical chemist. She's a chief scientist. She's led array deployments. She's a leader. She's training the next generation. So if analytical chemistry is your thing, there's a place for you in this program, the UNOLS fleet. Blake Watkins is an engineer that's also Terra Clemente. She's with NOAA. All things over the side. We do deep sea moorings, sediment traps, arrays. We deploy glass bottles with heavy water in it. We deploy radioisotopes on a string and recover them safely. That's pretty amazing. Blake has really taught back deck ops to a number of marine technicians in our program, to scientists such as me. I can even do these things every once in a while. An incredible teacher for the next generation. And I would be remiss without sharing the KM, OTG. Trevor's in this group. Everything and anything. You guys are the MacGyvers of the sea. You guys, every time we forget something essential, you just happen to have it or can make it or now can 3D print it. And on top of that, some of the marine technicians I've met in my career are just absolute wonderful people, and we've learned so much from you. So that's hopefully just a shout out and mahalo to all that you guys have done to make this science happen. So these were the guys that founded this program back when mustaches and short shorts were in vogue. Dave Carl and Roger Lucas, also Eric Fearing is in that photo as well. And I don't think they could have envisioned that this program would still be going 35 years later. And now we're deploying AUVs with ESPs to measure genomics in situ. We've got long-range instruments, we've got wave gliders, we've got sea gliders. These images of phytoplankton are captured by this thing called an imaging flow cytobot that we use machine learning to classify organisms. We are flying drones. We're still doing back deck ops in the way that done in the beginning, but the layers of technology on top of this are helping us understand the world in brand new ways. So I would say that together we are truly revolutionizing our understanding of the ocean. And the work that we're all collectively doing, like Anita has already pointed out, is helping us put together the pieces and understand how the oceans function and how they're changing habitability of our planet. And so I'm going to show some of the work that really has emerged from this program. And just keep in mind that every dot on this graph is a cruise that people have participated in to put together this composite view of how our ocean works. And what we are learning is that change is the only constant. These are temperature anomalies, so the difference from the climatological mean. We can measure that with satellites, we can measure that on moorings, we can measure that in situ, we can look at those anomalies from the sea surface to the sea floor. And we see strong decadal changes. So these decadal oscillations are changing the temperature of the entire water column in this ocean. Physics drive changes in nutrient inputs that change the rate of productivity. This is nitrogen concentration. These are low-level measurements. This is an oligotrophic gyre. These are very low nutrient concentrations. These are not simple measurements to make. They're quite challenging. But we found that mesoscale eddies and other physical perturbations are intruding nitrogen into the photic zone. That drives a pretty weak seasonal cycle. So even here in the subtropics, we're starting to see seasonality. That seasonality drives the abundance of organisms. These are Senecacoccus. This is a primary producer. This is a small 1 micron, so a millionth of a meter organism. It's quite abundant. There's 10 to the 6 per liter. And we're starting to see seasonal cycles of this organism, and we can start to see how that's changed over the 35-year time series. We also know the things that eat them. These are zooplankton. So when we're towing those nets and looking at the abundance of organisms, that's what goes into this data. For the first decade of this program, we had this increase in zooplankton that we could not understand. We still don't understand. And now it's sort of tapered off, and we're starting to see this strong seasonal cycle and abundance of these organisms that mirrors the rate of primary productivity. So this is that radioisotope using 14C, putting it in a bottle, tossing it into the ocean, and hoping we get it back safely. All satellite models, all productivity models, numerical models suggest that these oligotrophic gyres, where we are here, are deserts, and they're going to become less and less productive as we warm our planet. But in fact, our actual measurements are showing that productivity is increasing. There's a linear increase. We have some hypothesis about what's driving this, but this is really important because it helps ground truth these models of how our ocean works. And ultimately, that's driving carbon. So we deploy sediment traps, which you guys have seen these before. It's kind of amazing, right? You've just got a cup put out in the ocean, and you look at the rain of particles into this. It's essentially the rain gauge of the sea. And we can look at this carbon flux over time. We're also seeing this increase that mirrors what we're seeing in productivity. This is quite important because, at least in the oceanographic field, there's a large push at present to move towards what we're calling carbon dioxide removal, where folks are going to do sorts of things like add iron or change nutrient concentrations to increase this carbon flux, because this is the ocean's function of taking CO2 out of the atmosphere and moving it into the deep ocean. We're not the only time series in Hawaii. This is the iconic Keeling curve. If you get a chance to go to the Big Island, do. It's a beautiful place, and you can go and see this observatory. They've been measuring CO2 concentrations since the 60s, And that's what you're seeing here. You see that sawtooth pattern is a seasonal cycle. And now we're sitting at about 420 parts per million. The important thing to keep in mind here, so I was born in 1974, it was about 330 parts per million, now we're at 420. So the increase in a single human lifetime, not that I'm dead yet, but it's the equivalent to the change over thousands of years. So this change that we're seeing in my single lifetime is what we were seeing between an interglacial and a glacial cycle. This is a profound change. And it absolutely impacts the oceans. So in red, you're seeing that Mauna Loa Observatory. In blue, you're seeing the station Aloha time series. This is the Aloha curve. CO2 goes up in the oceans, it goes up, or in the atmosphere, it goes up in the oceans. And we're able to record that increase. So we see that same linearity, we see that same seasonality. And that change in CO2 drives a change in pH, the acidity of seawater. So we're seeing our oceans become less basic, more acidic, right? This is the process called ocean acidification. And this time series is one of many that's documenting that change in the most remote island chain on our planet. This is real, measurable, and persistent change. This is not a political statement. This is a fact that's drawn by data. So on top of the measurements that are our core measurements, we're also layering new technologies into this program. We've been measuring flow cytometry since the beginning of the program. This allows us to look at things that are a few microns in size, again, millionths of a meter. Now we're using optical approaches like forward scattering. We're also using imaging flow cytometry that allows us to make millions of images of plankton and again, use machine learning to classify these organisms. We're towing instruments called the VPR that again are allowing us to capture these organisms in larger size range. And we have a UVP on the CTD for every single cast that's taking pictures of things like diatom mats to mesopelagic fish. So now we can really, in real time, look at things from micron sizes all the way up to millimeter size and see how the structure of these ecosystems are changing in days to hours to weeks and hopefully into decades. Here's a few examples of images that we have from this. Look at trichodesmium, which is near and dear to my heart. We also look at marine snow, copepods, larval fish, diatom mats, and rhizaria. And again, it's in real time, and we're starting to build models that are allow us to classify this in real time, and on cruises, we can adaptively sample. And so ultimately, putting all these pieces together are really gonna allow us to address some grand challenges, right? We're starting to, between the moorings and the gliders and the remotely operated vehicles and our shipborne time series, understand how this ecosystem works, how it's structured in a genomic sense, how it's structured in a cell-based sense, and ultimately that allows us to look in this far right globe at this massive ecosystem that's the NPSG. We are one time series. Obviously, there are many. This is a paper that's about to come out. We stole somebody else's time series name, SPOTS. This is a synthesis product for Ocean Time Series. Each of the dots are a few time series that got included in this. And ultimately, by merging, by harmonizing data with the same methods across time series, we can start to get a picture of regional change, how regions are structured, how microbial systems are structured differently. And this is a little busy, but I want you just to point out the red lines. So this is a trend analysis. These are different time series. This is one metric. This is alkalinity, so the buffering capacity of seawater. And 3 points. One, Aloha is wildly data rich. That's the top plot over there, right? We've got a lot of data. This is one of the most data rich time series on our planet. And the second point is if we start to look at trend detection, right? So numerically, statistically, how much data do we need to be able to separate the signal from the noise? So for this particular property and other properties, 42 plus or minus 7, about 35 years. So 35 years, we're just now at the point where we can start to understand seasonality and long-term change. It's a massive investment in science, and we're just now at this point being able to statistically say that we understand the direction in which these ecosystems are changing. Why is that important? So, I've shown this in a few talks, but it's for me, it's pretty powerful. 3 to 5 years is a standard NSF grant, right? So if we were going to say, okay, I want to understand what the seasonal cycle of CO2 is in the oceans, right? You could pick one 3 to 5 year period and, okay, maybe I know seasonality. I can see how that amplitude changes. Go back 10 years later, okay, the amplitude's a little bit bigger, right? And then another 10 years later, and now you're really starting to see the fingerprint of change. That's nothing compared to what we have here. So these are incredibly valuable data sets that we've all contributed to. And so I hope that everyone who's participated in this program feels a little bit of pride in terms of knowing the amount of effort that went into these. So what have we learned? What are some major achievements at Station Aloha? It's a high quality, incredibly high quality, 35-year climatology, hydrography, productivity, export, biogeochemistry, and more. We have a gene catalog I haven't showed you, but 9 million unique protein coding genes, their taxonomic origins, functional annotations, and how those gene diversities change over time and depth. We have the Aloha curve that I've showed you, partial pressure of CO2, ocean acidification. And we have new views of microbial life in the oceans. Just a few years ago, we actually discovered a brand new organism in the middle of the subtropics, which is wild. That tells you how little we know about these ecosystems. What has HOT done for the University of Hawaii and really the planet? We have a conceptual understanding of the sea. This is the world's largest biome. We have actual records of ocean productivity and other ecosystem services. We have real measures of ocean carbon sequestration and climate, and some of the most accurate measurements of water mass properties on the planet. You would be amazed at how many salts we collect and run. It's wild. And we've also importantly provided access to the sea for volunteers, graduate students, scientists from a number of different institutions. So quite a bit. What have you guys done for us? Technology development, which is huge. We've learned an enormous amount from the work that's been done at sea. Training and education, it's reciprocal. We've learned, again, as much from the Martechs as you guys might have learned from us. Cutting edge technology. How else could we get to the bottom of the ocean, which is wild? And of course, access to the sea. Many of you guys have taught us many things in terms of how to do science. So I want to end with a little advice, unsolicited advice for the future. This is a self-repair manifesto. Like I said, I know you guys are all MacGyvers. I'm pretty sure I saw this on the Thompson. But I'll start with learn a programming language, right? You guys are here to do workshops and learn things. This is becoming mandatory for every bit of science that we do. Explore machine learning, AI. I was just at a conference, a workshop in Bainbridge Island. The speaker, a few sessions before me, was talking about how they were using the code that's actually behind Google Translate for people that had lost their sense of language. They were able to basically measure the brain waves of these individuals and allow them to speak, which is wild. And talking about the way AI and machine learning is being incorporated into our science, it's becoming everyday sort of work. So explore it if you get a chance. One of the things I absolutely love about going to sea is you get to meet people that come from different backgrounds and different places and really embrace your differences. I know the culture has maybe changed over time, but I think this is one of the things that I enjoy the most about going to sea is just meeting people from different backgrounds and being able to embrace those differences. This is the self-repair manifesto, something I've definitely begun to embrace. If you can't fix it, you don't own it. You guys all know that. We have all broken things at sea, and you guys have just the one part that we need to fix it. Make it an adventure. This is something I'll point out in just another minute as well. We get to go to amazing places, right? Most people don't travel in the sense that we get to as, seagoing scientists. So make it an adventure. And the last thing I'd say is be curious and keep your mind open. We have so much to learn from each other. So from the science perspective, we always want to see how the ships operate, what you guys are doing, what you're learning. So this is an adventure for us as well. And we also love for you guys to come and talk to us and ask us questions about what we're doing. And the last thing I'll say is don't forget to find beauty in life. Every time I think about the adventures I've had at sea, it really comes back to just how incredible this world is. And I'll just show a few. Many of you guys have been to the Antarctic. It's such a special place. It's absolutely amazing. I will always remember my time there. I studied trichodesmium for my PhD. Some of you guys know what that is. But basically, it means you're going to do your entire PhD in the subtropics in blue, beautiful places around islands. I highly recommend diazotrophs if you're interested in subtropical places. Lastly, this is Hawaii. I mean, it's such an honor for me to be part of this program. It's an amazing place. And I'm just going to end with this last one. Because, as I said, I'm really constantly amazed at the diversity of the oceans, the ways in which they function and sustain and support us. And so I urge you as you move forward in your careers to don't lose track of that bigger picture and how we all fit together and what we're trying to do here. We really are trying to understand how this planet works and how it's changing as a result of our own actions. So I'll end with that quote: "We will not starve for lack of wonders, but for lack of wonder." And thank you. I'm not going to take much more of your time, but thank you for believing, and I think the critical role of time series. We are sentinels of the sea. This is an image from us on an escape room that we did escape from. So thanks. Max. Use a comment. Yeah. The comment was just that the Hawaii Ocean Time Series is really the next iteration of the Keeling Curve, and certainly it is. We're looking at the ocean's role in absorbing CO2, and like I said, the MCDR stuff, the marine CDR that's coming down the line, which it will happen, I believe, is trying to enhance that sequestration of carbon. So dumping iron in the ocean, adding pipes, growing seaweed, burying it. Restoration of marine ecosystems, and all of it's trying to get at that flux of CO2, perturbing it even further. All right, great. Thanks, guys.